Thank you. Okay. Thank you, Tom, for the kind introduction. I'm here today to present the work of the Genetics Working Group. And the image you see here is one of a restored reef in Belize from Lisa Carnan, Fragments of Hope. And the goal of the working group was to provide guidelines on how to maximize the future adaptive potential of restored coral populations. The working group members include Andrew Baker, Sarah Davis, Andrea Grottoli, Carly Kinkel, Sheila Kitchen, Ilse Kaffner, Todd LaJeunesse, Misha Motz, Margaret Miller, John Parkinson, and Andrew Chance. And many of these members are here today, and they're going to be talking about the specifics of the guidelines this afternoon, starting at 1:15. This will be followed by a 15-minute discussion where you can provide us with feedback on how we can make these guidelines better. I would like to thank Tom and Tali for their tireless leadership over the last year and a half. I'd also like to thank Michelle Lowe for her logistical support and Steve Palumbi for advising the working group. There's many goals that we could have when we want to restore reef. Some of us want to restore a certain density of corals, others want to restore fish habitat. For the working group, we took an evolutionary perspective. So from an evolutionary perspective, the goal is to establish self-sustaining, sexually reproducing coral populations that have sufficient genetic and phenotypic variation to adapt to changing environments. So this was our goal, and this is how we wrote the guidelines, and we tried to be as specific as possible to provide guidance to the coral practitioners. And this, of course, increases the risk that we're wrong in some cases or others, but we felt that risk was worth taking rather than leaving practitioners without concrete advice. In the Caribbean, there's two main interventions we use for coral restoration. Crossing gametes to facilitate sexual larval propagation is one intervention, and the other is to fragment grow and outplant adult colonies to enhance asexual propagation. In the Caribbean, we don't have that many species of major reef-building corals. All of them are hermaphroditic broadcast spawners that are self-incompatible. Hence, you need at least 2 genets to allow for cross-fertilization and sexual reproduction. The other is that all of the major reef builders also fragment extensively. This includes not just the acroporids, but also the massives like Orbicella. And these guidelines are encompassing both of those interventions. First, the question is, which genets are you going to select to bring into your nursery or use for your restoration program? This comes under the topic of provenance, and Margaret Miller is going to give specifics about our rationale this afternoon. So, to enable the persistence of coral populations that are exposed to severe selection, not just from one, but from many stressors, we suggest that we need a mixed provenance strategy. So, we should collect genets both locally as well as from more distant, environmentally distinct sites. And we suggest that you need at least 4 to 6 genets from each of these environments to bring into your nursery. The reason for this is that our research has shown coral populations often have very locally adapted genotypes. So traditionally, the thought was that if this is the case, then local is best. The existing genets in your local environments are adapted to those environments, and that is what you should use for restoration. This is certainly the case to some extent, but the environments have changed so much, and our major reef-building corals are so old that they might have recruited many tens of years, hundreds, or maybe even 1,000 years ago, and the environment they're in now is no longer that way. Hence, there might be genets in other more distant environments that are now better adapted to these changing conditions, which is why we suggest this mixed provenancing strategy. There's many environmental gradients in the Caribbean, even though you might be more familiar with the ones that have been well studied in the Pacific, like Ofu Island or Palau, where we have an aragonite saturation state gradient. But in the Caribbean, we also have these gradients. We have high sedimentation sites and low sedimentation sites. We have inshore and offshore reefs. We have inner lagoon versus outer atoll environments. And so take advantage of those environments to source your genets and hopefully capture any locally adapted genotypes that way. The question then is, how many do you need? We reviewed our existing genetic data and calculated how many genets will represent the majority of the common alleles in your populations. And the answer for this is 4 to 6 genets should be sufficient to capture the majority of those alleles. For the specifics of this calculation, please go and see Carly Kinkel's talk this afternoon. Once you have selected your genets and brought them into the nursery, you're obviously trying to grow or put the most effort into those that have favorable traits. And we spend a lot of time in the working group figuring out what those traits might be. We've come to the conclusion that the existing phenotypic traits that are often surveyed are insufficient to really tell us this. So we came up with the following list of biometrics. This includes partial mortality, wound healing, skeletal growth rates, bleaching and infectious disease traits, as well as sexual reproductive output. And here specifically, we suggest sperm motility as a phenotype that you can survey with fairly little technology everywhere in the Caribbean. Once we have collected all these phenotypic traits, you can make decisions of which of these genets you want to keep propagating in your nursery. To leverage, however, this information on a broader scale, we urgently need databases that connect genotype and phenotype to really take advantage of this information. And we're currently working on trying to figure out how to establish such databases. It's not a small feat to accomplish. For sexual reproduction, we suggest that you need to combine the eggs and sperm from at least 6 genets into a batch culture. And this is because in our past research, these batch cultures with the contribution of at least 6 genets have higher fertilization rates, and they're more robust when you grow them, for reasons that are not entirely understood. The benefit of this approach is that you produce genetically diverse populations of larvae that now have new combinations of alleles that we can expose to selection. And those gametes are then settled and outplanted in both local as well as more distant sites. Now, we know that this rearing of larvae in captivity exposes or entails strong selection and adaptation to captivity. This is because these larvae grow in higher densities than out on the reef. They get pipetted, they get washed. So to minimize adaptation to captivity, your best bet is to shorten the amount of time these larvae spend in captivity. So where are you going to outplant either the larvae or the fragments that you've raised in your nursery? We suggest that you outplant both in novel as well as in historical habitats. And what we, again, as a research need identified is to get better habitat maps that allow us to predict which sites are now perhaps newly available for coral growth. So where are the sites that have now become actually amenable to coral growth where we should be outplanting these genets? We suggest that you tag and plant at least 4 to 6 genets in fairly close proximity, again, because from an evolutionary perspective, what you want is sexually reproducing populations. And our reef-building corals in the Caribbean often are asynchronous when it comes to spawning, so some genets spawn earlier or later than others. There's large compatibility differences amongst the genets, and sometimes genets forego spawning in a year altogether. So planting at least 4 to 6 together gives you some buffer in hoping that at least 2 will spawn at the same time in close proximity and produce genetically diverse larvae. You then again monitor biometrics of the outplanted genets and replace any poorly performing genets as necessary. Some of you may say, well, what about inbreeding or outbreeding depression? And without going into details, we have come to the conclusion in the working group that translocating genets among distant reefs is unlikely to be problematic from a population genetic perspective, but will likely provide substantial benefits. We also think that inbreeding depression is not a concern, given that current practices only raise first-generation offspring anyway, and that because our current populations are still so outbred, it is unlikely that you will by chance be selecting 2 very closely related individuals for breeding purposes. Finally, we considered the role of symbionts and in restoration. And here we come to the conclusion that manipulating symbiont during the fragmentation and nursery phase is unfeasible, and it's not likely that it will be particularly effective. Now, if you're raising larvae for restoration, the situation is somewhat different. It's easier to manipulate the symbiont populations, but again, we think your best chances of having a highly performing holobiont is to outplant those juveniles into local populations where they then take up symbionts from the environment that might be best suited to that particular place. The situation changes a little bit when we consider rare species. Here is a work of my graduate student, Annie Shan, who projected the future of Dendrogyra cylindris in the Florida Keys. And as you can see, there's many more colonies of Dendrogyra in the Keys than there are genets. And there's even fewer Breviolum Dendrogyrum strains associated with those Dendrogyra genets. Breviolum dendrogyrum has just been described as a new species. It's a specialist symbiont that we know so far only to associate with Dendrogyrum cylindris. So if this coral actually does reproduce, or a larva settles in the Florida Keys somewhere, it now may face a shortage, or it might be very difficult for these larvae to acquire Breviolum dendrogyrum from the environment, given that the adult population is now so low. And hence, that is one of the cases where intervention providing this particular symbiont species may be appropriate. This is the overview slide or figure from our draft guideline recommendations that sort of summarizes our thoughts. And again, I invite you to come to the session this afternoon starting at 1:15 to provide us with your feedback to make these guidelines better. So as a working group, we concluded that these basic guidelines should help maximize the adaptive potential of reef-building corals facing a rapidly changing environment. And sort of the mantra is curb carbon emissions and leverage existing genetic variation. Thank you.